Welcome back to our fourth video. Today, we're talking about adolescence. Adolescents are a group neuroscientists pay specific attention to, and for good reason. The adolescent brain is marked by a period of neurodevelopment where it strives to create a more efficient and precise working brain. The experiences and interactions during this time greatly influence how your brain becomes tailored to your specific needs, which is why adolescence can be such an influential time of interest for me and so many others studying the brain. The brain achieves efficiency in two ways: synaptic pruning and myelination, both of which will be covered in detail in this video. First, let's dive into pruning. Synaptic pruning is a developmental process in which unused synapses are eliminated and removed by non-neuronal cells called microglia. But wait, synapses are eliminated and removed? That can't be right. Trust me, it isn't as bad as it sounds. Let's provide some context to synaptic pruning. Time to go all the way back to the early postnatal days. During this time, rapid synaptogenesis is occurring. Synaptogenesis refers to the overproduction of synapses in the brain. During early postnatal development, we see an excess amount of synapses being formed. Synaptic pruning is a progressive reduction of this synapse number that continues into adolescence and adulthood. So don't think of this like we're getting rid of crucial synapses. Rather, pruning is a part of a developmental process designed by the brain to become more efficient. Mass-produce synapses, see what you need, and eliminate what you don't. This process is crucial. In fact, some evidence has shown that deficits in synaptogenesis to synaptic pruning process have been linked to developmental neurological disorders such as schizophrenia. Synaptic pruning is an experience-dependent process. Synapses not in use by the individual during the pruning period will be eliminated. This is what makes adolescence such a crucial time for brain development. The experiences and interactions you have aid in tailoring the way your brain is wired going into adulthood. Adolescents are frequently studied for this reason. Neuroscientists are using this life stage to investigate the cause and effect relationship of exposure during adolescence and the effects, if any, that come into adulthood. For example, using a model organism, such as a rodent, researchers could investigate the chronic exposure of nicotine during adolescence and the effects that may arise using behavioral assays and post-mortem analysis. This cause and effect relationship of adolescence to adulthood is actually what inspired me to investigate the impact of social interactions during adolescence, as we will cover in a future video. So far, we've established that synaptic pruning is a critical, experience-dependent developmental process. In addition to being experience-dependent, synaptic pruning is region-dependent. Synaptic pruning in the auditory and visual regions of the cortex is complete between ages 4 and 6, whereas synaptic pruning in regions involved in higher cognitive functions continues throughout adolescence. This means that the brain is maturing back to front, or posterior to anterior, with the frontal lobe undergoing synaptic pruning later in development than more posterior areas such as the occipital lobe. Synaptic pruning is the underlying process resulting in changes of gray matter volume, another significant point of neurodevelopment during adolescence. Gray matter consists of neuronal cell bodies and their respective axons, dendrites, and synapses, as well as glial cells and blood vessels. Gray matter volume and synaptic pruning kind of go hand in hand. When one increases or decreases, so does the other. For this reason, gray matter volume follows an inverted U-shaped curve during development. During postnatal synaptogenesis, gray matter volume increases. As the brain becomes more pruned, gray matter volume decreases, thus leading to the inverted curve. Like synaptic pruning, gray matter matures posterior to anterior. Gray matter thinning continues throughout adolescence in frontal cortical regions such as the prefrontal cortex. Okay, that about covers it for pruning. Let us now turn our attention to another crucial part of the adolescent brain development: myelination. While synaptic pruning is the removal of unnecessary synapses, myelination represents the addition of material. Myelination refers to the process in which axons are wrapped in fatty cells. This addition of insulation allows electrical signals to travel faster in the brain than axons that are unmyelinated. How? Instead of signals traveling continuously along the unmyelinated axon like we see here, myelination allows for the signal to jump along the axon. These jumps occur at points called nodes of Ranvier. Nodes around VA are short regions in the myelinated axon that are not insulated by myelin. These nodes contain high concentrations of voltage-gated sodium ion channels that amplify the action potentials at each node via the influx of sodium ions. This acceleration of the transmission of action potentials is another contributor to the increased efficiency we see of the brain during adolescence. Just like synaptic pruning, myelination typically occurs posterior to anterior in the brain. Sensory and motor areas are completely myelinated around preschool years, whereas higher cognitive regions, such as the PFC, are not fully myelinated until late adolescence or early adulthood. Like we mentioned before, synaptic pruning and changes in gray matter volume kind of go hand in hand. We see a similar relationship between myelination and changes in white matter volume. White matter mainly consists of bundles of myelinated axons that allow for the communication between different areas of gray matter. The most famous example of white matter would be the corpus callosum, which is the white matter responsible for connecting the right and left cerebral hemispheres and is the most prominent white matter structure in the brain, consisting of roughly 200 million myelinated axons. The volume of white matter increases throughout life until early adulthood due to the progressive myelination of axons by myelinating cells called oligodendrocytes. Changes in white matter volume is more linear than that of gray matter volume and tends to be less variant across brain regions. All right, I just threw a whole lot of information at you, so let's just quickly recap. Synaptic pruning and myelination are both ways the brain becomes more efficient during adolescence. Synaptic pruning is an experience-based reduction of unused synapses, which results in reduced gray matter volume in these regions. Myelination is the process of wrapping axons in fatty myelin sheaths, creating nodes around VA that allow for the faster transmission of action potentials along the axon, consequently resulting in an increase in white matter volume in the brain. Both processes are key to neurodevelopment. Adolescent neurodevelopment serves as an important time for the individual where the brain undergoes adjustment to help one function best given their external environment. The developmental plasticity seen during this time provides the opportunity for the brain to be impacted by the activity and experiences of the adolescent. Prior research has shown that our social brain regions of interest mentioned in a prior video undergo changes during adolescent development. These areas include the affective network, amygdala, orbitofrontal cortex, and ventral striatum, involved in emotional processing and regulation, the cognitive control network, the VLPC and DLPFC, involved in impulse control and higher order cognition, and the social cognition network, TPJ, PSTS, DMPFC, MPCC, and MPFC, which has been implicated in the evaluation of social norms and adjustment in personal behavior to conform to these norms. These neural changes are essential for the improvement in social functioning and are crucial to the development of social species. That about does it. Thank you for tuning in to this video highlighting changes in the adolescent brain. See you on the next one.